Hello, everyone, then. Can you distinguish large riparian sedges? I wonder. By riparian here, I'm including both those that grow on the banks of rivers and those that grow on the shores of lakes and in allied habitats like canals. But they are a very interesting group of plants ecologically, but there are some issues with them which I will come to in a minute. I did consider an alternative title for this talk, which was How Do You Join the Ranks of the Most Revered Botanists? And the answer is, get one wrong. If you've made a mistake in this area, you are in very exalted company. And indeed, as you know, there is an old proverb that if you make no mistakes, you make nothing. Well, my interest in this group goes back to this man, Hef Warburg, who was my tutor at university and is also very well known, of course, as one of the authors of what used to be the standard work on the flora of Britain and Ireland, Clapham, Tooting and Warburg. Now, in 1966, as I was about to start on a research degree, he suggested that there were some unresolved problems in the section of Carex that we know as the acuti in those days, now known as Phacocystis, and that they would be a good subject for a research project. That is the group of which the Irish representatives are Carex acuta, Aquatilis, Bigelowii, Elata, and Nigra. So I spent 3 years researching that particular group, which ended up with a particularly work on experimental taxonomy and on breeding, various interbreeding studies. Now, enough of that. I have done no research, serious research on sedges since 1970 or thereabouts, but I have maintained an interest. And then by about 2010, 2012, something curious came to my mind, or I began to realize that Carex acuta, a plant I was very familiar with in the late 1960s, is a plant I had never seen in Ireland. On the other hand, I had seen Carex aquatilis a number of times. And if you look at the Atlas 2020 records, the map of Carex acuta shows a very considerable scatter of dots around the country. The paler dots are the pre-1950 ones, the darker ones are the 1950 to 2000 ones. And there are only about half as many dots for Carex aquatilis. So in all, Carex acuta was in 87 hectads, Carex aquatilis was in 39 hectads. Now, since the number of hectads, this is a back of the envelope calculation, but the number of hectads in Ireland is about 1,000. So if acuta and aquatilis were randomly distributed in relation to each other, you'd expect the number of hectads which had both species to be 3.9% of 87. 8.7% of 39, if you like, which is the same figure. Right, so that would work out at about 3 hectads with both species recorded in them. On the other hand, I know that it has been suggested that these are two species which replace each other geographically. In other words, they're allopatric. And if they're fully allopatric, that means you would find no hectads or thereabouts with both species recorded. Well, how many actually do have records of both species? And the answer is 19. There's something going on here, isn't there? Is it that they both like the same sorts of place? Or, perish the thought, could it be that the same population has sometimes been identified as one and sometimes as, that is, as acuta or sometimes as aquatilis? Well, they do look a bit the same, don't they? Well, I thought I'd better go and have a look at some of the sites for where a cuta had been recorded and see what I could find. And what I found is summarized on the right-hand side. And now I will stress that so far this is work in progress. I haven't been to all the sites yet, and most of the sites I've looked at in the northern half of Ireland. But what I found was in some places, there are now no sedges present at all. In others, there was Carex aquatilis and no acuta. In others, there was Carex Cutiformis and no acuta. In others, there was Carex nigra and no acuta. And in some that I came across, although I think these ones I talked to others about rather than visited myself, was Carex Riparia and no acuta, or Carex elata and no acuta. And very rarely, I did actually come across some Carex acuta at last in both counties Down and Meath, but nowhere else so far. Now, it is apparent then that there is an issue here. We seem to need rather better recording of these large riparian sedges. And that is what this talk is about, mainly how to distinguish them rather than about what you will actually discover, because there is more work to do on finding out what their distributions really are. Now, features of riparian sedges then, the list on the left here. These are the species at the edges of rivers and lakes, and by tall I mean sort of more than about 60 centimeters when they're full grown, a bit longer than that perhaps. In general, these are long-lived plants. They perennate by rhizomes, all except one spread laterally by leading rhizomes. And sometimes they form dominant stands, or even monospecific stands in some cases. When it comes to growing in shade, they survive perfectly happily, but they tend not to flower in shade. And a few, one other point, as compared with, they're all in the subgenus of Carex known as subgenus Carex, which are the ones that have a terminal male spike and maybe some more male spikes, and then the female spikes are lower down, and they look quite different. The other thing I would say is that for the most part, they are self-incompatible. They do require cross-pollination. Now, I have drawn up a table of some of the characters, and I'm not expecting you to read this and appreciate it fully. It's still work in progress, but I hope to have it complete in a few days' time, along with the associated notes. And it means nothing without the associated notes, I'll stress that. So if anybody was interested in getting a copy of this table, let me know, or let Paul know, and I will forward you a copy as soon as it is complete. But there's a number of characters down the left-hand side which are critically important. And I want to have a look at some of those because it is quite evident that they are giving trouble. Firstly, and this is probably the easiest of them, is whether they are whether they have long pioneer rhizomes or whether they're tussock forming. And most of you will be very familiar, well, some of you anyway, that have studied this kind of habitat, we're very familiar with the tussocks of Carex elata or elata, as shown on the left-hand side here. And they're quite different from the stands of the even kind of stands which the rhizomatous ones form. Just occasionally, you will get an in-between stage, which you can see here. That is what looks a bit like a tussock, but is a very wide spread out tussock. And the reason for that is there's just actually a hybrid between the tussock forming elata and the rhizomatous Carex nigra, which is probably the most common hybrid that one will encounter in those. And that's in I think in a Turloch in County Sligo. The even type stands there are depicted on the left. This is an even stand of, I think it's character Cuta. And just picked out here is is an individual stem. It didn't come from that population. It's taken earlier in the year. Again, this is character Cuta, a shoot with a long rhizome out here, which is probably 25 centimeters in length, something like that. If you grow them in pots, which I often do, then these rhizomes sort of go round and round the pot, which is quite distinctive, very easy to tell if they're producing that sort of rhizome. So it's interesting. It's important to look at the actual growth pattern of the plant. Now, a critical character, though, is the number of stigmas. And I think this is one of the ones that gives most trouble. It's very easy when you start early enough in the year to look at the plants and you see the stigmas as they are here coming out in pairs from each flower. Here, there's one, there's another one, and you see they sort of diverge slightly at an angle. Very easy to say that there are just two stigmas there. A little bit later on, and it's still quite apparent that there are two stigmas. But the stigmas become a completely entangled mass after a while. And if you're looking a bit later on at that stage, it can be extremely difficult. Now, unfortunately, I don't have a slide of an entangled mass, only of a stage in a different species. This is actually Vesicaria, where they are beginning to fall off. And here you can see one there that looks as though it's got three, one there that looks as though it's got two stigmas, another three up there, a possible two there. And what's happening here? But the reason is, of course, that one stigma may have fallen off, leaving two. So if you count two at this stage, you do not know whether there were three there or whether there were two there originally. And I think what has sometimes happened is that people may have been only able to count two when there were actually three. So that is something to be very aware of. There's no simple way around it, except that in this particular case, you've got a great inflated utricle, and that is a clue I'll come to in a minute. Just male spikes are also worth looking at. And here you have, this is a male spike at the end of the inflorescence, and on the shoot above, you can see a male area at the top of a female spike. Now, what is apparent here is that the anthers are dehiscing much later than the stigmas become receptive. So they're protogynous. The female part matures first, but also that you've got these little terminal stretches at the end of the upper female spikes. So worth looking at carefully, because the nature of the male spikes is quite important. Here we have a Cutiformis, a Cuta, and a Quatalis. And they're all quite neat looking spikes, but the Cutiformis ones tend to be chunkier, whereas the Quatalis, later, Nigra, all the rest of them have these rather neat elongate spikes, narrower in dimensions across the width. But the Cutiformis is rather variable in that context, so it's not entirely reliable. But here we have one which is usually pretty clear cut. This one is Riparia with large brown chunky male spikes. And not only are they large, brown and chunky, but they appear relatively early in the year. Flowering time actually does distinguish these species, but not without overlap. Riparia and Elata tend to come a little bit early. Indeed, looking at the elater growing on my pond yesterday, I could see that one of the two tussocks growing in the pond is already showing its male spikes. And this is still only March. And I wouldn't be at all surprised if you were to go to the Newry Canal where this picture was taken, you wouldn't find some male spikes of the Carex riparia already evident. Whereas the others, elater, acuta, nigra, are sort of middle of the road ones, and Aquatilis tends to flower a little bit later. But you cannot depend totally on flowering time if you've only got a small population or you've got a population odd situation. The next character to look at is the Utricles. Now, really, there's only one that stands out very clearly, and that is Riparia, which has a much longer utricle with a prolonged beak and these 2 points, it's a really bifid tip to it. A Cutiformis, if you read the books, will appear to be a little bit different, but it isn't at all easy. It's ribbed, it's a little bit longer, it has a notch, but again, there's overlap because there's so much variation within each of these species. And here you've got a typical nigra, almost negligible beak up here compared with a short beak in a Cutiformis. And most of the other group, elater, acuta, Aquatilis, are all somewhat similar to nigra, at least on average. Next character that is, I think, an underrated one, but very, actually, I find it very useful, is to look at the bases of the sheaths, the leaf sheaths. And this one is Aquatilis, and you can see a distinct reddish sheen off these, which is, in this group of sedges, pretty well confined to Aquatilis and a Cutiformis. But you can also see in this particular shot that the shoot bases are fairly rounded. They're not very acutely angled. And that is also characteristic of Aquatilis. The shoots tend to be rounded at the bottom, and these trigonous stems that all sedges have are quite bluntly angled in Aquatilis compared to the very sharp angles that the other species have. Now, another character to look at, and I think it's not shown terribly well here, is the color of the leaf surfaces. This one is Aquatilis. Most of them, the color that is fairly similar, but in Aquatilis, the leaf surface underneath is dark and a little bit shiny, as opposed to somewhat glaucous above. And the glaucous look is usually due to the presence of lines of stomata. Maybe not terribly clear there, but if I go on to a much closer picture, this is actually of the Cutiformis, then you can see these white lines of stomata quite clearly here on the underside of Cutiformis, whereas they appear not to be present at all in the upper side of the leaf. And now, there are two species where the stomata are largely on the upper side, and they are Aquatilis and nigra. I was hoping to include a slide of Aquatilis yesterday, and I went and picked a leaf in the pond where I have some Aquatilis as well, put it under the microscope, and lo and behold, I could see stomata on both surfaces. So it wasn't obeying the books. Maybe the books are only right in the middle of the season. So I thought I'd better show you one that actually did obey the rules. But I think that point does illustrate it's a guide, but treat it with care, the distribution of stomata. Where else? Now, bract length. This is critically important. And it's the length of the bract in relation to the inflorescence that is key. This one is elater, and here you can see this bract here is quite short. All of these bracts are quite short in relation to the inflorescence, and that is characteristic of elater, although they are somewhat variable, and I have seen them where they reach about the length of the inflorescence. This is nigra, also quite short, shorter than the inflorescence here, and that is typical. That's the edge of a lake with nigra going on it. 10 yards down the lake shore, more nigra, rather more erect plant, but this time the lower bract is exceeding the inflorescence. That is not so typical, but it does happen. Be careful, because that is only Carex nigra. It is not one of the longer species, which one of the taller, larger species where the bracts normally exceed the inflorescence. Like this one, Carex acuta. Here you can see there are at least 3 bracts, 1, 2, 3, which are as long or much longer than the inflorescence. And in the case of acuta, they tend to arch over the top of the inflorescence in a rather graceful way, which you can remember because the old name of Carex acuta was Carex gracilis. Gracilis, gracile, graceful. A rather lovely sort of architecture when you see it in a classic form. You also have very long bracts in Carex aquatilis, and there is something else to note about this as well. Firstly, you will see the tips of the leaves look very glaucous. Don't be misled by that. That means very little. It's very common in these riparian sedges to have the tips of the leaf looking very glaucous. Even the ones I picked in the dark last night out of the pond had this glaucous area at the top. Anyway, this is typical aquatilis in that there are about 3 bracts that exceed the inflorescence. But there is something else to note here too, which is that the female spikes, and these ones are atypically short, I'd say, but they're very closely enveloped by the base of the lower bracts. They really almost ensheathe and surround the inflorescence at one stage. And the angle between the bract and the rachis of the inflorescence, now, where's that? That's the, sorry, my...
John, can I jump in and say you've just got 2 minutes?
Okay, that's all I need, I think.
Okay, that's good.
This angle in here or there is a very narrow angle. And that contrasts with a Cutiformis, where the angle between the bract and the female spike tends to be wider. But as I have used the word frequently, tends to be wider. You'll see that there's a leaf down here. I don't know if that top bract actually subtends another inflorescence, another spike lower down, and is maybe forming a much narrower angle. But I certainly see that angle as often being something which gives me a clue that we've got a Cutiformis present. So just to sum up with these large riparian sedges, of which I've got another specimen on the right. That's my favorite one. That is Pseudocyprus. It's a really gorgeous plant. But yes, some advice here. Beware of poor specimens. If you're going to do measurements, make sure you're doing it on a typical measurement, a typical inflorescence from the population, not on a little runt somewhere in the corner. Remember that heights and leaf widths and leaf lengths are plastic things. If you've got vigorous plants, they will be taller and wider and have longer bracts in relation to their inflorescence than if you have little runty things. The stigmas have to be counted with care, as we discussed a few minutes ago. You have to remember to look at the leaf sheath bases, and that's very easy to forget if you're collecting something in the field. Oh, I'll just snap off of an inflorescence and maybe that will help me. No, you must get down on your hands and knees, get your knees wet, and look properly at the sheath bases. Look also at the male spikes as well as the female spikes. How many are there? Are they neat, or are they chunky, and are they loose? Look at the leaf surfaces, and you can take a leaf specimen back and look at it under a microscope, but you can usually see the stomata with the lens perfectly okay. And also look at the leaf or stem section. One character I didn't mention here was the leaf section. Most of them are very plicate, but in Quaterlis, there's a tendency for the leaves to be like a sort of broadly spread U in section, shaped like a bit like a house roof gutter. But again, not always, but that's a fairly good character that I would use. And if you're in doubt, please admit it. Don't record whatever don't just have a go and say, oh, I think it might be this. You know, take a good look, and if you're not sure, well, record it as uncertain or send a specimen to somebody else. I'm very happy to comment on any specimen sent to me. And if you would like a copy of the table, let me know by email. Thank you.